Hello, we are live.
Yay.
I'm
Hi.
gonna actually just post it on my Blue Sky too while
Oh,
we
yeah,
just be like,
I
"Hey,
totally
folks,
forgot
we're
about
talking."
social
Yes.
mediaing.
Uh-huh. I put it on Twitter and my Instagram today. So
I totally forgot to blue sky.
I'm just gonna be like, "We're starting now."
Yeah.
Yeah. So anyway, hello. We were just
Hello.
talking about how we've both been experiencing like surprising. Oh, hey, Saeed Simeon. Um,
Welcome back.
surprising. Yes, welcome back. We've been having some surprising successes in our research. Like Ruby and I were both like, wow, we were doing stuff and we're just like, wait, what the heck? That actually worked.
Yeah, it's a
Wait,
good week. Yeah, I was like, whoa, that worked. We need to repeat it
uh-huh.
to
And
make
that
sure
was
it's not a
that was
fluke.
that was imaging some cells.
Yeah.
Yeah.
We were imaging some brains, and we kind of like I was gonna not cowboy science, but kind of like I'm gonna just try this, and maybe it'll like help us verify it. And then we saw it, and then we're like, no way, we got to do it again. So now we have to wait like 3 weeks before we can see it again. But it's very promising, which is a
Nice.
good place to be. But yeah, this week we thought we would oh sorry, let's do the intro so people know who we are. Hi, my name is Ruby.
I'm
Welcome,
Nathan.
and yeah, welcome to Blossom Sciencing. And this week we wanted to go back to our roots. Like back in like pandemic era when we started the stream, Nathan and I would just like have random zooms once in a while, and we would share random cool facts from the news about science with each other, and it would boost our mood. And I thought that we should just go back to some of the things that really did well for us then, and hopefully you guys enjoy it. So we each prepared a couple, or more than Nathan did more than a couple cool science news facts, and we thought to share with you. And I actually haven't heard his facts, and he hasn't heard my facts.
Mhm.
So and feel free to stop us and ask questions. So yeah, I thought I want to hear the cool things that are happening in the science world that Nathan learned about this week.
Sure. So this one was like almost a cool thing. Um, I'm at the Media Lab and we actually launched some instruments on a mission to the moon recently. Um, so it was one of the first kind of privately funded, well, not privately funded because it's funded by NASA, but privately operated lunar lander missions. It's uncrewed. It's just a robotic system, but it was actually designed to explore the landing site where Artemis 3 is going to be landing. So Artemis 3 is going to be the first human mission coming back to the moon in 2026 or 2027, and it's going to be landing on the pole. So this mission was going to go there and scout around that area and especially look for water, ice, and things they could use to get resources from. And actually, so the Media Lab had like a couple different things they were testing on it. One was a thing for scanning the environment and then rendering it in virtual reality. And another one was a small mini robot that could crawl around on the main rover and inspect equipment on it. So for like troubleshooting and diagnostics. Um, so we built actually this like mission control center in the Media Lab, and it was really cool. And then like the rover landed, and it like tipped over, so they couldn't do anything with it. But also somewhat disappointingly, like this company has launched two rovers to the moon, and both of them landed and then tipped over. So like they're just not very good at this, I guess.
Oh, that's so upsetting. I actually walked through the lobby of the Media Lab fairly recently,
Uh-huh.
and I saw that there was like multiple mission like goals,
Yeah,
and one goal was just to like get there. It was like it
right.
had like a disc with like
Uh-huh.
information, like was it like music or like culture or
Yeah,
art or something?
like music and like messages from different people. So it's kind of like if you know like the Voyager Golden Record thing, it's like a message to outer space.
Yeah,
Yeah.
that part made it
Yeah.
intact, we think.
Uh-huh. Yeah. No, like what so one of my lab mates worked on that part of the mission. And so she was like, so she was like, well, my mission worked. It's on the moon. And sucks to be these other people who need to like do stuff on the moon.
Oh, that's really funny.
Yeah. But yeah, it was pretty cool because they actually they actually built like the mission control for these like missions in the lobby. So the idea was that like when people were working there, you could just like kind of walk by and see it, and they do like informational stuff when they were planning activities. So it's kind of sad it didn't get to work.
Oh,
But
that would have been so cool. So like the actual like
Mhm.
people
Yep.
controlling the
Uh-huh.
rovers would be sat like in the lobby.
Yes. Uh-huh.
That's
Yeah,
really
yeah.
cool.
Yeah.
Uh,
Yeah, so it's sad that it didn't work, but it was a really cool way to do a moon mission, I think. And to like educate people about it and show how it works and stuff.
oh, that's so cool. I would have loved to just like hang out in the lobby while this was
Mhm.
happening because it would have happened like 24 hours, right? Like
Right, yeah, it was actually like quite depressing because everyone was just like standing around there and then they realized it wasn't gonna work. But uh,
Ribeye says the first line in my to-do list is to make a to-do list.
nice.
Then you can cross it off.
Nice. Make
Like
to-do
me,
list. Like got it. Yeah.
making it. That's
Uh-huh.
great.
It's made. Yeah. I guess I can mention a cool thing that actually did happen, um, which is that we managed to essentially cure someone of type 1 diabetes. Um, and the way this works is so type 1 diabetes is an autoimmune disease where your immune system destroys the cells that naturally produce insulin. And so in this patient, what they actually did was um, they use this technique called induced pluripotent stem cells, which basically you can take a different kind of cell, like a skin cell from someone, and you can genetically reprogram it to be essentially the equivalent of an embryonic cell that can differentiate into any kind of tissue. So you take cells from an adult, you basically convert them back into embryonic cells that can then become any kind of tissue, and you then reprogram them to become these insulin producing cells and put them back in the patient.
So,
Um,
did they like inject those cells into the pancreas? Like back
um,
where it's supposed to be or like
so some researchers that have used this technique have done this. In this patient, they actually put them in her abdominal muscles. Um,
wait,
and
so
they
she
said
just has a bunch of mini pancreases growing
uh-huh,
in her muscles?
in her abs, yeah.
Like, okay.
Um, and they said the reason actually they talked about the reason that they did that was because when they're closer to the skin, it was easier for them to measure their activity. They said using
Oh.
MRI. So I guess that so I think like something about how you position the coil, maybe you can get like a higher signal to noise ratio when you're closer to the skin. And also had something gone wrong, it would have been easier for them to take them out.
Oh. Wait, I
Um,
never thought they would put it, like, just under their skin and their abs.
mhm.
Like, I thought it would go at least,
Like
like
back in the pancreas.
yeah, or, like, internal, like, inside, like, the like, not in the muscles, like, I thought it would go into the cavity, like,
Oh
all the
yeah,
way
just like
inside.
somewhere in the abdominal cavity. Oh yeah.
Yeah, like hanging out
Um,
by a liver or like, in your guts and stuff, but actually it makes sense if it's like, "Oh my god, it's going haywire, let's just chop it off."
uh-huh.
It
Yeah.
makes
Oh yeah.
sense
Siren
to leave
Simeon
it.
says, can you link the article? I just dropped it in the chat. Yeah, and but the cool thing is that it seemed like this worked. So what they measured was basically they measured her ability to maintain kind of a stable level of blood glucose by herself, like a healthy person could. And people who have type 1 diabetes, like normally cannot do this at all. And her ability to regulate her blood glucose levels steadily improved and got actually to the point where she needed very, very little treatment
Oh,
after she had this done.
that's that's great
Yeah.
because I feel like one of the issues with diabetes is that you actually constantly have to monitor if you're high or low. And like if you have something in your system that naturally has brakes and like levers to make it go and stop
Hmm.
naturally, um like all you need is just the functioning cells again. Like your body can
Uh-huh.
tell you like, hey, stop making so much insulin, like you haven't eaten food in a while, or like you just ate a lot of sugar, like bump up your activity, go. Like it's
Yeah.
kind of cool. I mean, it makes
Exactly.
sense.
Yeah.
Yeah, yeah.
It's like I guess these cells like have like they also just inherently sense blood sugar and then they regulate like it's just these cells can both sense it and then produce insulin. So you just have to put the cells somewhere and
Very
they'll
good.
do their thing. I will
That
say
is so
one
exciting.
yeah, I'll say there is a big caveat to this, which is that she also does have to be on immunosuppressive drugs. Um, and the reason is because if because type 1 diabetes is a autoimmune disease, if you
It
just
would
put the
attack
cells
its
back,
own cells
like
again.
it would just kill the cells again. Um,
Wait,
and I'm
but someone who has type 2 diabetes where they don't have an autoimmune thing that kills their own own like insulin making cells, they
mhm.
don't they would just have to like put shove some of this their these cells in their
Yeah,
abs,
yeah.
right? Like it would be like
Yeah, although
it
like
could work, right?
I think normally they have normal insulin homeostasis too. Like their insulin levels are responding normally and it's just the cells their other cells don't respond to the insulin is the problem.
Mm.
So these pancreatic cells well, I guess these pancreatic cells would be churning out lots of insulin because their blood sugar. So maybe you could just make people like maybe just you increase their total ability to make insulin and maybe that helps a bit.
Yeah.
Like
Yeah, or they would just like have a stimulator that like activates
mhm.
their production like when they consume food or whatever. But overall really
Yeah.
exciting. Like transplanting your own cells back to well like yeah,
Uh-huh.
like your skin cell that became a a stem cell that became a like a islet cell like back and forth and then
Right.
transplanting back to the people like this was like some
Like
sci-fi like dream
yeah. I mean
like
it's pretty cool.
sequence.
I mean this
Yeah.
has been thought of as like an incurable disease for a long time. And so I think it's like it's really promising and
Oh,
it makes a lot of sense. I'm also really curious about kind of what happens like so they're not putting them in the pancreas, they're putting them in the muscle. I'm curious about like what that means for like muscle performance too. Because it's like the insulin is being released basically directly onto the muscle cells. So those muscle cells are actually getting like very, very high concentrations of insulins presumably.
yeah, that's true. Now I'm also wondering like what they told this patient to do. It's like you must not do any crunches. You must not like aggressively
Uh-huh.
use your abdominals.
Yeah, because like you change like the sugar in that area, and then it does like causes this burst of insulin.
Yeah, it's
Yeah.
sometimes it's wild that things work at all. Or like
Uh-huh. Yeah. It is
like
one of those, yeah, things from just like, wow, that worked. Yeah.
yeah.
Oh yeah.
Oh,
Oh,
that's such a
Science
cool one.
Simeon said like what they did with the MRI was very cool.
Oh,
Uh, what did they do with the MRI?
yeah. Let's Google or
Mm,
actually let's just read the paper.
yeah. It's this trans why
Paste
is
that.
MIT not letting me access it? Mm,
Download full PDF. Here we go. Here we go. Oh, I'm
oh,
just
there we
reading
go.
newspaper.
Wait, do we not have access to Cell at MIT?
We
This is
don't have
weird.
access to Cell. Cell is a Selvier.
Oh, right. Yeah. Hmm. Oh man, this is the thing that we were talking about last time.
Yeah, paywalls.
Mhm.
Uh, Yeah, I actually have
Oh, I found a
access.
PDF though.
Okay, great.
Um, so this is what scientists do.
Oh, Science Simeon says so they were looking for water and fat signals that were specific to the normal growth of the cells around the expected transplanted area, and that everything wasn't growing out and deforming. I suppose you don't really want like a mass, like,
Mhm.
and, like of
Like an extra pancreas that is just kind of like sticking out.
like a glob, like a lump.
Mhm,
That makes
mhm.
sense. Oh, so
That
it's
makes sense.
really like integrated into the abdominal muscles. Like it doesn't even stick out.
Mhm. Yeah.
That's
Which, like,
wild.
I am very curious if you did this in, like, your leg muscles or something, like, how would it affect your exercise performance?
Yeah. I like that we went
Uh,
straight to exercise performance.
mhm.
And it's like, are they superhumans now?
Yeah, I mean, like, you could imagine that it could, right? Because it's like then you have something that is directly in the vicinity of those muscles that's regulating how much glucose they can take up. Um,
Yeah. Oh, wild.
like because like when you're exercising hard, you do have local hypoglycemia in your muscles, which is why you kind of well, I guess is it hypoglycemia or is it lack of oxygen? I don't know if which is actually the limiting factor, but
Yeah, but now I'm thinking about growing other things besides pancreases. Like I feel like like you could grow things that would make your muscles perform better, right? Like you can foresee like you could grow cells that would make a bunch of like things that could shuttle out lactic acid, for example. And then
right.
like you can imagine this person would not get sore from like
Mhm.
intensive crunching and like and
Yeah. Or
I
like what if you put like androgen producing cells and you just like put them in the muscle so like the total systemic amount of androgen is very low but it's like being released directly onto the muscle cells.
yeah. Evie Neural says like you can grow more neurons to your
Uh-huh.
abs
Yeah. Oh yeah,
or
Revive E says testosterone, so we were thinking about certain things, similar things. Yeah.
yeah.
This neurons idea is actually interesting. So, like, this is a thing that I learned recently, which is when you are doing strength training and the first phase of muscles getting stronger is actually all related to nerve growth. So, what's actually changing when your muscles first start getting stronger is just that your neurons essentially grow more synapses and get more effective at making the muscle fire. So, presumably something that released like neurons or neurotrophic factors could actually make you a lot stronger.
Yeah, no. Okay, now it's like this is like a new level. Like there was like doping, and then now there's like grown different
Cell
cells.
doping?
Yeah.
Yeah.
Now it's like I mean, I guess blood was cells too. It's
Mhm.
literally myoglobin and
It's cool
hemoglobin.
though,
Yeah.
like if you can make things that have kind of these homeostatic loops built into them though, like
Yeah.
so that you can
You
like
don't
implant
overdo
something
it.
right.
Yeah.
Implant
Okay.
something that's
Yeah. Now I'm worried that you like implant something and then it'll just like go haywire, which actually is a great segue into one of the topics I prepared because I prepared cancer.
nice.
So like we're like, we don't really want a giant ball of like pancreas growing like
Mhm.
in your abdominal muscle. Um, but so I actually saw this paper come out like kind of like just peripherally, and I was like, "Oh, it's kind of interesting." And then you're like, "Prepare something," and I was like, "Actually, let me read the paper," and I
Mhm.
actually kind of liked it. It's kind of unconventional data gathering. So like you usually think
Huh.
about data gathering like you have a lab, like you grow a bunch of animals, you collect data from it, and you really want like the animals to be like siblings, you want them to be the same
Mhm.
age, you want to control like all the things. And this cancer study was like we want the opposite of that. We want the most number kinds of animals. We want them from all across the board. We want them from everywhere. And we just want to look at the prevalence of cancer across the animal kingdom, and they're like, we're gonna go to like all the zoos, because at zoos they have to do necropsies when an animal dies,
Mhm.
and like every animal has a necropsy result, and when they do necropsies they can like look for masses all over the body. And so
Mhm.
that's exactly what they did. So they went and looked at, okay, I wrote down the numbers. They analyzed 16,000 necropsy records from like 292 different vertebrate species. So the question was, like, is cancer prevalence, like what is the cancer prevalence in like different types of animals? And there's this like long going paradox that was called Peto's paradox,
Mhm.
where we like, okay, so if you just imagined cancer being like a problem, a mutation that happens every time a cell splits, then theoretically an animal that has more cells should have more cancer, right? Because there's more
Mhm.
opportunity, like just like lottery, there's
Right.
just
Yeah.
more chances of getting cancer. But then he was just like, but look at a human. Like human is like 1,000 times bigger than a mouse and has 1,000 times more neurons than a mouse. Why is there more cancer in mice than in humans? Like, why
Right.
isn't there like
Like,
astronomically
why
more
why
cancer?
do humans not get cancer 1,000 times more than mice do?
Yeah, and
Yeah.
we talked about this back when we were like super animals or something. We talked about how like
Mhm.
elephants like don't get a lot of cancers, and like they had these like redundant like telomeres that were like just super long, and it was just like it was really cool. And we're like, wait, hold up. This like contradicts that so that's the paradox. The paradox is
Mhm.
like, why are these big animals not having more cancer? But the problem with that is that we knew about elephants, we knew about mice, and we knew about humans, and like maybe like two other species, and we didn't really have like a full like breadth of animals to
Of,
look
like,
at.
all the animals, yeah.
Yeah, like just like
Mhm.
go across the kingdom, everything that has vertebrae, like let's just compare them, like the frogs, the monkeys, like everything in between. And so that's what they did. And I guess like surprisingly, actually the paradox is not true. Like that paradox is
Hmm.
only for the
Mhm.
animals that we had known. It actually they couldn't find a good correlation. Um like they did find a correlation between size and prevalence of cancers. Like they couldn't
Mhm.
find like the opposite version of that. And like it wasn't a
Right.
super strong um like correlation. They actually
Mhm.
like you could only really find it like when they controlled for like gestational time. So actually the longer the time you are gestating, like those species
Mhm.
that have like longer gestation times have less cancer, or like less
Huh.
like neoplasia cancer, which is just like
Uh-huh.
the tumors, not like the malignant kinds of cancer, just
Mhm. Just
like
like any
one.
kind of tumor.
Yeah, like massive tumors. And actually I wrote down the exact, like I did bullet points because I knew I was going to forget something. So large animals tend to get more neoplasms and malignancies when compared to smaller animals. Longer gestation periods predict lower neoplasia and cancer prevalence. Species with fewer somatic mutations also developed less neoplasia, which
That
like
makes sense,
matches,
yeah.
yeah. So like our original hypothesis, or like the null hypothesis for most people, is actually true. um but they also noted that like the evolution of cancer susceptibility appears to have like undergone different shifts, like sudden shifts where like certain like clades of the animal kingdom would like suddenly have like huge shifts, and then it would become like pretty stable afterwards. So then they were like, also here are your outliers, here's our weirdos. And so I wanted to talk about the like super non-cancer animals and then
I
the
guess,
super
like,
cancerous animals.
yeah, I guess I was just wondering, like, before we get into that, so, like,
Yeah.
so if you control for gestational period, you do see an association between body size and rate of cancer?
Yeah.
Okay, yeah.
Yeah.
So they're saying, like, there is no paradox
Yeah.
as
Paradox.
long as you control for gestational period for some reason. Okay,
Yeah.
yeah.
Yeah. And more DNA damage means more cancer. And like
Mhm,
I feel like we kind of knew that, but like
mhm.
it was this weird paradox where we're like, why are these big animals not, all keeling over with cancer all the time and
And the
dying
answer
young?
seems to be because they have long gestational periods?
Yeah, it's because we didn't control for the
Mhm.
gestational period, and so that seems to be protective. So it's not just like, okay, death with this paradox, like it doesn't make sense. It actually kind of opens up a like like an avenue of study. Like, let's go
Uh-huh.
see what gestational what happens during gestation that like protects cells from like going haywire later on when they're dividing.
And like, yeah, I mean, I guess the thing that, yeah, so Science Simeon is saying, what is it about gestational period? I guess like that intuitively makes sense to me because it's like, okay, if you have factors that just limit how much cells can divide, like presumably then you have lower risk of cancer, but then you can't grow as fast in utero too, right?
Yeah.
Like,
Yeah, and actually, like there's like some evolutionary things too. So like for example, like amphibians had the lowest medium neoplasia prevalence across like
mhm,
compared to like mammals. Like the amphibians in particular, like kind of like an old ancient like a like proto-brained animals. Those guys had less cancer overall ish than mammals. Like why? Like what
mhm.
about being a mammal makes us more cancerous? Like
I
more
mean, we
likely
have a
to.
faster metabolic rate, right? Like we're producing more free radicals.
Yeah, okay. So that's where the outliers are interesting. Okay, I wrote down
Mhm,
the outliers because I wanted okay, so let's start with like the super low cancer rate animals, which is super
mhm.
surprising. So a fruit bat, which is
Huh,
a
uh-huh.
bat, like which eats like a lot of food and
Makes
flies a
sense.
lot.
Fruit prevents cancer.
But high metabolism, super small
Oh, I
animal,
see. Mhm,
and like like it's like it's a mammal, super
mhm.
low cancer, like super
Right.
low
Uh-huh.
cancer prevalence, like less than 1.6. Whereas on the other end, a also small animal that's a mammal, like the ferret, which has the highest prevalent, one of the highest numbers. They had like 63% prevalence. Like that means like more than half of the ferrets that
Get
died of
cancer.
cancer. Yeah. But okay, I actually like oh no. Oh no.
Oh.
I
Oh, no.
this
Do
room
you wanna
forgot
get
I
up and
was
turn
here.
your lights on?
Yeah, no, it's motion censored. Okay, wait. So I was just like, what the heck is going on with the ferrets? And so I actually texted my vet friend and I was just like, hey, what's up with your ferrets? Like, what's going on? And she actually told me something kind of cool. She told me that a lot of ferrets came from one source. All ferrets in zoos come from
Mhm.
one place. So she said, okay, they come from a common source, Marshall Farms, and so the cancer likely bred into those lines, and because they're so inbred,
Huh. Uh-huh.
um, like they're small and they live a long time. Um, so domestic ferrets have high cancer rates, and so it's just like, yeah, I think those ferrets that they were studying at the zoos, they weren't wild ferrets. Those were
Uh-huh.
like farmed ferrets, and so that likely like skewed the results. So that was
Yeah,
kind of
Revive B says, like, there's just one guy breeding all of the ferrets.
it's kind of true. Like there's like I forget like a couple years ago, like one company bought all the naked mole rats like that are like the whole colony, all the colonies of naked mole rats. If you work with them, like and you
Mhm,
purchase them, they basically come from this like one or two colonies that they
mhm.
just bought all of. Okay, sorry, we're missing a whole bunch of there's
Oh,
stuff
yeah.
happening in the
Yeah.
chat and I've totally missed it because I was like
Yeah, so
talking about.
basically, so Science Simeon was first talking about just the paradox and was saying like, so yeah, if you have more division events, then why would it be so crazy to think you have a higher likelihood of cancer? So like, essentially, that's what they found, right?
Yeah,
So yeah. Mhm.
it's logical. It follows logical sense.
But it's like
um
we had other when we were just looking at like a few other types of animals, it was not clear that that was the case.
Yeah, and it's
Yeah.
likely because mice is not a representative mammal,
Mhm.
and neither are we. Like
Yeah.
we're not a representative mammal. Like
Do you think the mice have the same problem? Like if you're looking at laboratory mice, just their cancer rate is all weird because they've been
oh,
selectively bred or
let me maybe I'll look up that. Oh, actually, that's probably true. Like some
Mhm,
animals. So actually, if you go and order mice, and I've done this before. Like I've actually walked up to a vendor and they have like they call them outbred mice, but
mhm,
they're like different varieties of strains, and they all have like different tendencies. Like this one
mhm,
has huge litters. These, are more likely to be like like singing. They're better at singing. These are like really jumpy. Like these are like, do you know that they have like
Yeah, yeah, yeah.
it's kind of like when people are like have dogs and they're like, these dogs are
Mhm.
dumb as doornails and these are smart dogs. Like this is
Yeah,
the it's kind of like that. And generally true. If you like meet enough of them, you're like, yeah, actually those border collies are real smart. And and these are doornails. Um okay, ribeye says
yeah.
make more
So
baby
then, like,
snacks.
yeah, so then Science to me was also saying, like, oh, yeah, there's a big confound, like, in that these different animals maybe have different amounts of inbreeding or different kind of artificial selection pressures placed on them, which, yeah, I mean, I feel like I've had lab rats and pet rats, and I think one of the interesting things is like domesticated animals like actually do get lots of really weird kinds of cancer, usually just because it's a side effect of like the things that you were selecting for, or because you weren't selecting against cancer. Like I think it is really easy to through artificial selection, like accidentally mess with the risk of cancer. Um,
Yeah.
um,
Yeah, I think, I mean, if everyone wants advice, like mutts are probably the best.
mhm.
Like most likely to live the best or longest lives with the least amount of problems. Or they
Yeah.
could just inherit all the problems. But like
Yeah.
um
And then so there's
we
also
could
this
do a whole thing on dogs.
discussion, yeah, there's this discussion about like, okay, what's the evolutionary benefit of like getting rid of cancer protection that Revive was asking, which I guess like one argument could be like, well, it lets you have a faster gestation period, maybe. Like, so if you like we know, for example, like a lot of mammalian cells actually kind of have built-in limits on how much they can divide. You get maybe like 40 or 50 divisions out of them, and then they just stop. And we think that's probably partially an anti-cancer mechanism. But the downside is it really limits how much you can grow or how fast you can grow, and also how much you can regenerate.
Yeah, I
Um,
bet you it's a speed thing. Like, I wonder
yeah.
if we could just like compare cell division rates. Like how
Hmm.
many, like, do you know what I mean? Like how
Like if
many
that
times you can
predicts it.
divide. Yeah,
Yeah.
yeah, like in the same amount of time. And I think I mean it's encoded in our genes. Um
Or also just how often do cells become senescent? Like I wonder it's like because like we have all these checkpoints where certain things will happen and cells will just kind of stop dividing and it's kind of an anti-cancer emergency switch sort of thing. And I wonder like animals where cells kind of hit that checkpoint and stop frequently, maybe they grow slower and they also have less cancer.
yeah.
Yeah.
Um Okay, sorry. I was telling people about the
Noise.
animals. So the animals with the lowest prevalence of cancer are the porpoise, the common porpoise, the fruit bat, and the black-footed penguin. And on the flip
Huh.
side is the ferrets and the possums, coming in at 63% and 35% for the possums. Like those are the outliers. Like those are what those are
Mhm. Like, honestly, I'm not really seeing a pattern here.
no.
Like
No, and if you want to look at the paper, we will drop it and also drop it in the chat. Did I drop it here? Eek.
um,
Copy. Am I dropping in the chat?
it doesn't seem to be dropped.
I failed at dropping in the chat.
Oh,
Nathan
do you
shall drop
wanna
it
I
in.
can try and drop it.
Yeah, well, you can drop it. Yeah, so there's
There we go, I see it,
the
yeah.
article. Cancer animals. Yeah, I don't know why that like really like tickled me that day. I was just like, you know what? I'm gonna read this paper about animals across the animal kingdom and cancer.
Mhm, like, cancer prevalence across vertebrates. Well, I mean, it's a really interesting topic, especially given that there's this paradox that people have talked about.
Yeah,
Um,
paradox no more. Which is great. It's kind of like like science changes when there's new information, and here's like a whole buttload of new information. Um,
mhm.
Thank you all the zoos and all the technicians and vets and whoever else is doing necropsies around the world for 16,000. Like this was like a meta analysis. Like I feel like coordinating the data would have been really complicated. It was like, can
Mhm.
you send me all your
Like
data
getting
on your
all this data from zoos. Yeah.
yeah, like different zoos, like all the different kinds of paperwork from different zoos. It's like, yeah, can you give me all your koalas and your like penguins, like everything they have? Wild. Anyway, I think we have time
Let's see.
for a couple more.
A few more?
Yeah,
Yeah,
you want to do one?
sure. Um, yeah, so one of the things that I was reading about are some studies about GLP-1 drugs. So these are the drugs that, like Ozempic, kind of these weight loss drugs. And I was reading some really interesting studies that are coming out that were suggesting that not only do these seem to help people with weight loss, they also seem to be helping people with addictions. And so one of these studies that came out that I'll put in the chat was actually they were taking one of these drugs that's called Axenitide, and they gave it once weekly to people who had alcohol use disorder. And what they found was that that actually seemed to help compared to a placebo. So, in addition, it was an add-on to kind of their traditional to their kind of typical therapy that they were doing, but half of them got this drug, half of them got a placebo, and the people who got this drug had fewer binge drinking days than the people who got the placebo. So obviously, this is very strange because it's like, why would this drug that like regulates appetite um, help reduce drinking, like help with alcoholism? And one thing that's actually have been suggested maybe is that it seems like in studies in rodents and also in people that one of the things these drugs might be doing is modulating dopamine responses to natural rewards. So there's been some studies in rats that suggest that maybe one of the mechanisms for how these work is that they actually kind of reduce the amplitude of the dopamine response to eating highly palatable foods. And that's part of kind of how they make animals eat less and have less appetite. And that also maybe has some benefits to kind of helping animals or helping people get away from using these really highly addictive substances like alcohol.
I was gonna say this seems like this is where not alcoholism, like addiction and like alcoholism like overlap with food over consumption, like
Uh-huh.
overlaps with alcoholism is addiction, right? Like these are probably similar circuits that are being driven,
Yeah.
overridden, over incorrectly doing their thing,
Mhm.
or it's
And
just
it's
like
like,
evolutionarily in our system to keep us hunting for food so we can
yeah.
die off.
And I guess one thing one thing that like I kind of wonder about this, and this is like just like entirely me speculating, is like, okay, is it that this just kind of has a direct effect on dopamine signaling, or is it like just if you make people somewhat less hungry, are they less vulnerable to other psychological problems? Like that's kind of the like one thing that and like the reason I speculate about this, I was talking about this with some friends. It's like if I'm very hungry, I actually feel lonely. Like for some reason, like I misattribute that. And like I'm like you can say like, okay, maybe there's some theoretical connection, but like I wonder how much does just reducing hunger help people who have like alcohol use disorders or addiction or impulse control disorders or something like that.
Yeah, that's true. I don't know. I did know that like these drugs, Ozempic, like these GLP-1 drugs are doing so well that food
Mhm,
companies are actually like making food, like they're changing like the foods, like they're changing your snacks to make sure
mhm.
that like they're still addictive even
Oh
if
wow,
you're
even
on
if
the
people aren't
drugs.
hungry.
Yeah.
Although, like this research would suggest maybe that's not going to work though, right? Like if it's acting on these central reward mechanisms, then.
yeah, then maybe
maybe,
it
yeah.
doesn't matter, right? Like even
Yeah.
as long as you're on these drugs, like
Mhm.
you won't be addicted to food
Yeah.
at all. Um,
Ribeye B says the HALT in Alcoholics Anonymous stands for hunger, angry, loneliness, or tired. So these are, I
ah,
guess, factors that cause people to drink.
ah,
Yeah, yeah. I mean, given that, like, it does seem plausible. Like, okay, if we know that like hunger is associated with um, alcohol use, and you make people less hungry, um, there are. I will say the other weird wrinkle of this is that like it seems like this is also involved in health in a lot of weird ways. So actually, one of these GLP-1 drugs just got approved as a treatment for sleep apnea. Um, and in that case, it seems like the mechanism is just so like being overweight is one of the biggest risks for sleep apnea. So in this case, it seems like it's just making you lose weight, and therefore it's uncompressing your airway, and you can breathe easier. Um,
I
but
like that.
also,
That's approved, like for like a secondary. Like it's like you're treating a symptom that is really not the actual disease.
right,
Or
like it's like
you're-
yeah, like it's like you're like the sleep apnea is a symptom of being overweight. So to treat the sleep apnea, you make people lose weight.
skinny. Yeah,
Like,
I mean, if it
it's
works, it
if it
works.
works, yeah. And actually, I mean that's kind of the really interesting things about these drugs is that like we actually know now it's like okay, they really seem to be kind of helpful for everything. Like people who take them tend to have better heart health. They tend to have kind of um, better sleep, like we were talking about. And like the thing that I wonder about is like, okay, is this all just because kind of it's improving people's basic metabolic function? Like, and I think that's really plausible. I think like
Ribeye.
maybe the mechanism is just yeah, put it in the water.
That's. I feel like there's an ethical thing here.
I mean, okay, but hmm, if we're gonna fluoridate the water, is it that different from like GLPing the water?
That's true, but does everyone need it? Maybe all of America needs it.
I mean, the weird thing about like, I feel like from a public health perspective, you could actually make the case that the majority of the population would benefit from this.
That's true. I mean, historically, we've done that, right? Like, there's iodine in
Mhm,
our salt. There's like micronutrients and minerals
mhm,
in our cereals, like for this reason, right? Like, we have done
yeah.
this in the past.
And
I
also
f-
expense. So like the reason we started putting fluoride in water as opposed to just like in toothpaste and other things was because it allowed it to reach people who didn't have enough money to get that kind of regular dental care. And similarly, the GLP drugs that we have right now are really expensive if you're getting them like the regular injectable form. So it's a similar argument. It's like, okay, yeah, put it in the water. Get it to everyone.
that's true. And then ultimately, preventive care is saving money because now they're not
Hmm.
going to end up in hospital with like more horrible diseases later in life, right? So,
Hmm. Yep. Uh-huh.
like, that is like
I
the
I'm
home
not sure
goal.
if I'm not sure if you're joking, but I'm somewhat convinced of this now.
I'm gonna go look up the side effects of the GLP-1 drugs now.
Honestly, I think that Revive is just like, yeah, I it is like I feel like most of them are not super orally bioavailable is the hard part, but um there are some companies making like GLP-1 gummies now, so maybe you can do it.
Yeah.
Um,
Uh, actually, this is a surprisingly good segue to to my topic, which is actually involved with creating proteins, which is
Ooh.
um like if you could create a protein that has a functional, like that can do whatever you want it to do, like GLP-1,
Mhm.
like these drugs, which are proteins. Imagine you could just designer it Do you know what I mean? And then you can like
Mhm.
produce it really cheap. Like if you could
Mhm.
fold it, if you could cause it to basically be the exact shape, functional shape that's useful, you could do that. And that's actually the other topic I prepared today, which is about protein folding and how AI has really advanced um, our ability to predict the structure of proteins, and also allowed us to basically things like DALL-E, like which is basically
Mhm.
ChatGPT for art. It's like draw me a duck, riding a horse, riding a
Mhm.
frog or something like in a pond. Go. And like it could generate you this image. But you can imagine if the prompt was draw me a protein that has like the reactivity of Ozempic, like like
Mhm.
one, and it needs to be cross reactive to this. It needs to be this size. It needs to be. Do you know what I mean? It needs to cross the blood-brain barrier. It needs to do this, and it, like, and then it just like goes, and it gives you like 20 iterations of it, and then you make it and you try it. Like, what a cool new way to like make new drugs.
Uh-huh. So people are doing this now,
Yeah,
or yeah.
it's actually what's happening now that we have so much access to like AI engines and okay, actually I want to go back like history because
Sure.
like that's kind
Mhm.
of my thing. Let's start from the beginning. So historically, it's really difficult to know um like what the protein shape is going to be just by what the amino acid chain is. So like you can go from like DNA to like RNA to protein, like just amino acid like a chain. But from
Mhm.
amino acid chain, you can basically kind of sort of just predict like alpha helixes and beta sheets. And you're like, yeah, from the hydrogen bonds and from like the electrostatic, it's going to be like these shapes. But after that, it's really hard to know how it's going to fold, and it's actually one of like the biggest problems in biology for like the longest time. And it's such a big problem that actually, let me put numbers. I actually wrote numbers because i I homeworked. uh okay, so like this is like a big, like a huge problem. Let's see, so let's say you had a amino acid chain that was like 30 amino so sorry, a protein chain that was 30 amino acids long, and from that the number of possible folds is 10 to the 33rd power.
Mhm.
So even if you had the most like strongest computer that could do like 10 or 20 different iterations of the folds like every second, it would take you like longer than longer than 100 times the time of the universe that has existed
Mhm.
already to fold all the different versions and test if it was like the most stable structure. So like just brute forcing it is not doable.
Doesn't
Like
work.
yeah,
Yeah.
it doesn't
Mhm.
work. And so historically, the way we've like figured out the shape of proteins is we used X-ray crystallography. So basically, you like you go find this protein. Like the first protein they found was figured out the structure of was myoglobin, and they had to like go get a hunk of whale from like a deep diving whale because they have a lot of myoglobin, like
Oh. Hmm.
get the muscle, like basically extract out all the proteins from it and like make a crystal of myoglobin, like a big crystal of myoglobin. And then from there they x-rayed it and then like got the diffraction pattern, and then like mathed it back to its like original shape, and it's like super fancy. This won the Nobel Prize in 19 I wrote the year 1962. This is for John Kendrew's 1962. If you ever want to look it up, just look up the turd of the century.
Yeah.
That is the name of the structure of myoglobin. It was the first time anyone saw the structure. Science Simin says, crystal of myoglobin was not my bingo card today. But yeah, look up the turd of the century and it'll show up. um and so like that took a lot of work and it was like a huge deal. And basically from then on, like 1960s to 1980s, they've been like slowly picking off like one protein at a time, and they're like really careful about it. Like this needs to be a really important protein for blank, and so we're going to figure it out because it causes diseases and stuff. And so they were picking them off, and so in like the next 20 years they figured out like 100 protein structures. And like so like by the early 90s they were like, this is like way too slow, like we need to use computational So like, we need to do like we got to use computers on this problem. And so in the 1994, so like a couple years after I was born, some scientists were like, let's have a competition. Like if we make it the competition, like
People
any
will do it.
yeah, people will do it. And so like every year or every 2 years, I think it's like biannual, they would like release a like a new prompt basically. And so they knew the final structure, but it was hidden from everyone. And they just gave them the amino acids. And it was really simple. It's like, here is like a string of amino acids, go. And go predict the closest thing. And whoever got closest to the actual structure wins. And they did this for like a lot of years, and basically and then the goal was to get like 90% accuracy. They're like, if you can predict the structure up to around 90%, like that, like you basically predicted it. And no one was there, like they were like barely performing at like 20%. um and so they so some of the forerunners was actually something called Rosetta Stone, which I actually is where actually I first learned about this problem. So when I was a kid, I don't know if you know about Rosetta, like it was like this screensaver. It was just I thought it was just a cool
Oh
screensaver.
yeah, the folding. Yeah, yeah,
Yeah,
yeah.
the protein
Hmm.
folding screensaver. So basically the idea was like we don't have supercomputers, so we can just socially engineer like everyone to use their own computers, download was it Rosetta Stone or
Rosetta at home, right?
yeah,
Yeah.
Rosetta at home. Yeah. Download Rosetta at home and you'll get a cool screensaver. But it like so you get a cool protein screensaver, but that protein screensaver was actually everyone's computer running all the iterations of
So
protein
it's
folding.
basically a botnet that
Yeah,
they.
it was like the OG botnet. It was happening in like libraries. It was happening like I had that screensaver at some point in
Mhm.
elementary school. It was like everywhere. So that was kind of cool. And that performed around I think it like was 40%. It was like pretty
Mm.
good.
Mhm.
So this like biannual thing was called CASP, by the way, and CASP stood for Critical Assessment of Structure Prediction. So it was like a competition every 2 years. Um, oh yeah, it capped, but then it started to plateau around like 50% accuracy. Like it wouldn't get better. It couldn't get better with like every year. Every year, it just like consistently performed around 50%, and it just never like
Mhm.
made it over the hump. Um,
Right.
but like some people that were like watching their screensavers because they're just like staring at it and they're like, that's a stupid fold. Why would you do it like that? They were like, we could do better. Like they're like, okay. Then they were like, let's make a video game. And so they like made this video game called Fold It. And I played so much Fold It in high
Mhm.
school and college.
I'm just
Like
amused that people are watching their screensaver and they're just like, "Why are you doing that?"
like that's a stupid way to do it. Because essentially it was
Mhm.
brute forcing it, right? Like
Mhm.
Rosetta was basically brute forcing it, but using like every library computer that was
Mhm.
idling and like just making it do the thing,
Mhm.
which I feel like is kind of a good use of like that time.
Yeah.
Um so anyway, that capped out, and so they were like, Let's see if like humans can do better." And indeed, within like the first like 3 weeks of dropping Fold It, the game, the video game, like 50,000 video gamers like hardcore played it, and they like solved the structure of like an HIV enzyme. Like, and they were like,
Wow.
later like we found the structure of this enzyme that's super important for this virus, and like they're like cited. They're like one of the authors. Like, it's like Fold It
Mhm.
Gamers. Like it's like
Mhm.
one of the authors. We'll drop it in the chat. I think it's like one of these right here.
Yeah, I can. Let's see.
Yeah, so 50,000
Video game.
gamers pooled their efforts and they solved this enzyme, which is like I was just like, wow, citizen science, right? Like these are just people like sitting at home. Like I downloaded Fold It onto my like sad like university laptop, like the monster laptops, like with the square screens from that era. I remember doing that. That was really cool. So anyway, fast forward to DeepMind. They were like, let's try it with like the best computational power we have on the planet right now, which was Google's DeepMind. They're like, let's use these computers, let's push it. And they put teams together and they really we can do a whole AI episode on exactly how this worked. But basically they used combination of information that we knew before, like what kind of folds we typically see with like these different kinds of amino acids. They also added information about like the evolutionary information. So like they compared across animal species, and then they trained it with all of the data from the currently known proteins, and then they
Mhm.
like did a bunch of cool like loops. They use this thing that like ChatGPT uses, which is like like the attention part of it.
Mhm.
We can really like delve into
Yeah,
this more deeply in
AI.
a
Yeah.
separate episode. But like
Mhm.
basically they used AI, and the really cool part of it is that okay, I wrote the numbers down because it kind of blew my mind. The numbers were exactly, we went from so in 60 years, like before the AI like came on, before the AI attempts at this, we figured out like 150,000 genes, and it took us 60 years to do 150,000 genes.
Like to figure out what the protein they made was
The
shaped like.
structure was,
Yeah.
yeah,
Mhm.
like that was how many structures that the all of science in all of the world has figured out. We figured out approximately 150,000, and it took us 60 years. And as soon as AI came on, so this is around like, I don't know, 2010 to 2020, like this paper came out in 2020, they solved 200 million. They solved 200 million structures. That is essentially most of the proteins that we know in nature. They solved all of them in like in a really short amount of time. It's wild. Like it's like 3 orders of magnitude more, like
Mhm.
in like a fraction of the time, which is really cool. That's a huge leap forward for science. And so it's almost to the point where now we can just make designer proteins. Now we're just like, yeah, we know these proteins should be like this. Can you make me a protein that looks like this and does this, and specifically is like this, and then like go like
So it
go.
works bidirectionally too. So you can say like, okay, I want something that has this shape, and you get like the gene sequence, like the
Yeah.
genetic sequence you need to make it. Yeah.
Yeah, so pretty much that combination. So like one combination was like figure all the structures out, and then the other combination
Mhm.
was like how to like make your own proteins. Like it's okay. So traditionally before this, it's really hard to just be like imagine a protein shape. Like look at like the shape of the myoglobin, right? Like the turds of the
Mhm.
century. Like be like, like let's make let's make the shape of blank. It's really hard to just like draw one out without like staring at a lot of them and like using the collective knowledge of all the possible shapes that already
Yeah.
exist. But this is exactly what AI is really good at, right? It's really good at
Yeah.
taking things that they kind of know before and just like, you know,
Uh-huh.
um fixing it up. Like it's large this is like large language models,
Mhm.
like they're this is their thing.
Yeah. And I
So
will say, like, I think it's important to emphasize like how hard this problem is, because like this is really like the key to really actually designing a lot of therapeutics is, for example, like you know um for example, like this virus has a particular receptor that it needs to replicate, and if that receptor is blocked, it won't replicate. So you want something that will make that will stick to that receptor. But before this, we didn't really have the ability to do that. Like, so
no.
what you would have to do is you'd have to like find some organism that like preys on that bacteria and makes something to disable it, and then figure out how it does that.
Yeah.
Or you'd have to kind of like just tweak a bunch of things until you found something that worked, basically.
Yeah, no, that's exactly how we do it. So like for
Mhm,
example, like venoms. So like let's say you got bit by a
mhm,
snake like in the wild. Like the way they make you an anti-venom, like or actually the way the anti-venoms work right now is like they collect the venom from like a whole bunch of snakes, and then they're like anti-venom A, anti-venom B, anti-venom C, and each one of these anti-venoms have like a bunch of different species of snake venoms in them that are like most prevalent. Like if you're in the southwest, there'll be like southwest snakes, and then they'll collect all those venoms and put it into a batch, and then they'll inject that into like a horse or like a cow or some big large mammal, and then collect the serum from those animals, and then like isolate the antibodies, and then they give you those antibodies. And
mhm.
so like you always end up with like a bunch of extra stuff. You can become allergic to it. You can become you can like your body can like learn to not fight them anymore. And then like next time you get bit by a different snake, it's not going to work anymore. Like you only get like one chance with those like
Mhm.
like venom, anti-venom A from the Southwest. So like if you
Yeah.
can't get multiple bites from an animal, basically, like it can really like the anti-venoms will cease to work. So you can imagine, because now it's so much easier to design an anti-venom antibody that you can just like supplement one. You could literally just on the shelf be like rattlesnake A, rattlesnake B, rattlesnake C, like every single different kind of rattlesnake, we got you an anti-venom specific to that snake. And so you're like, what
Mhm.
snake bit you? And you're like, well, it's this one with the orange rings and the black things. And like, okay, we think it's that rattlesnake. We'll give you that specific anti-venom. That's so cool.
Yeah.
Like we
Huh?
couldn't do that before. Like we could
Right.
just
Mhm.
invent the antibodies that it's supposed to have, and it'll just make
Yeah.
you just make it, because you know, once you know the amino acid sequence, you could just we can make amino acid sequences really easily. Like I think it's like 50 cents a amino acid right now. Like if you just order illegals like that.
Mhm.
So like as long as we know like the specific sequence, we can make it. Um yeah,
Mhm.
what a cool way to
And
use
so
AI.
like yeah. And so like if I have a protein that I like so like if I want to make a protein with this, so I would use this AI to get a gene sequence and then like would I like put that gene sequence in yeast or like how would I get the protein?
Yeah, you could grow it in yeast, you could grow
Mhm.
it in culture, you could
Mhm.
probably
Although
I
presumably like some bacteria like can't really make like human proteins very well, right? Like
no,
because
I
they're
think you can. Yeah, no, I think that's like the basic of most synthetic stuff is you just make it in cells. Like you can make it in
yeah.
like I think it might have to be eukaryote cells. It probably really
Yeah, like
depends
some
what kind
need
of proteins they
eukaryote
are.
cells because they have to be like factors for it to like fold properly. But
Yeah, but like it's
mhm.
kind of exciting. It's an exciting time. Like
Yeah.
uh post
Oh, Revive
translational
says post
modifications.
translational modifications.
Oh, that's true. I don't know about that.
Mm.
But hey, but you could make it the final form, the form that you want
Right.
after the post translational modification. We just make that. We just skip that step
Yeah, like you could just make a protein that doesn't need any weird stuff to stuck
to
to
like
it
fix
for
it,
it to
right?
like work.
Yeah,
Yeah.
because like the idea with proteins, it's really the structure and the shape that matters for function, right?
Mhm.
So it doesn't really matter how it gets there. Historically, evolutionarily, it's like if it ain't broke, like don't change it, right? So they'll start with some version of it, and then like they just keep tacking things on top or taking things away until you get like something that does the thing better than another thing that does the thing. Like we could just pick the thing that does the thing best
Mhm.
from the get go. Um, what a cool thing! Like
Yeah, that is really cool.
yeah,
Yeah.
it's really it's such a huge leap forward, like, we don't have to be like, "Oh, what is the structure?" Like, it used to be, like, the structure of one thing. That's, like, someone's whole PhD. Like, these 15 people plus a supercomputer, like, all the tensor chips and whatever of deep mind Google, like, computers, they did it in, like, a couple years. We're like,
That
"Wow."
would be really depressing if you were working on doing this using other methods.
Uh,
Like,
I
it's depressing for all the crystallographers
well, no, so they still use a crystallography to make sure it's right.
to verify it.
Yeah, to
Yeah.
verify it. So, like, it's like once you get really close, right? Like, even decreasing
Mhm,
it down to, like, 10 versions of the closest protein it should be, then you just check it with the crystallography. And also crystallography is really expensive and takes a lot of time, and it's like, you gotta, like, X-ray shit and you gotta do math and stuff. You can reduce the number of like, you can't brute force X-ray crystallography, like, it just takes a really long time. So, if you
mhm,
can, like, reduce it to, like, the lower number of probability like, possibilities, you can just check like, cross-check and be like, "Alright, look at that, like, you're over 90% accurate." And in 2020, they, like, exceeded the CASP, like, gold
mm,
star record of 90% accuracy, which is
yeah.
very cool.
And
Um
I guess one question I have is, like, so why is this computationally feasible? Because like, we talked about, like, how it would take impossible amounts of time to do, like, brute forcing it. So is the idea just, like, there's actually a bunch of rules about protein folding that we haven't discovered, but like the AI has kind of implicitly learned, and that narrows down like the possibility space. Or
Yeah, so we can go in deeper exactly how the
Mhm.
AI does it, because there's
Yeah,
not a lot of
I mean,
time.
I guess
But
I'm mostly just interested in like what does this mean about protein folding? Like
oh,
why?
okay,
Yeah,
yeah. So
like it
brief,
seems like the fact that it's predictable using AI means something, right?
yeah,
About
yeah,
like we were missing something about how this process actually works.
yeah. So like if we sat down with like pen and paper, we could make a bunch of little predictions. The
Hmm.
nice thing about using AI for this is that they can summarize a bunch of these little predictions, and then like so if you imagine you took all the information about biology,
Hmm.
like these kinds of hydrogen bonds are usually more likely to be next to this hydrogen bond, and elephants and humans are more similar than elephants and reptiles. And like you can like you can put all those kind of biologically phylogeny tree, you can put all that information in one spot. Then you have like another spot which is like trained data on all the proteins that exist. And
Hmm.
then so you basically loop them. So like all these, like every time you add a new animal, new facts about stuff, you would take that and you loop it into the trained data set with the like all the known proteins and structures that are already known. Then you take information from here and then you loop it back into, you add it to the model here. So you basically make two parallel models and they're constantly feeding each other. And basically that's how they let it intelligently teach each other. Like it's like a two-way teaching
Hmm.
model. And then that's how they generated like the possible structures.
The structure, yeah,
Yeah.
yeah.
um but I can do more homework and tell you
Yeah.
exactly how they did it. But that was like the general gist of it. I think it's actually
Mhm.
a really cool AI story, and maybe we'll bring
Yeah.
on someone that studies AI to talk about
Yeah.
this.
We were thinking of doing like some AI streams actually. Like
Yeah.
I think actually one of the things we wanted to do was talk about like AI art and how does that relate to like how human creativity works.
Yeah. Um I don't know. This is AI art, right? Like
Mhm.
this is art and science coming all
Yeah,
at
I
this.
mean it is like a creative activity if you're like, hey, design a protein that docks to this thing. Like,
Yeah,
yeah.
like docks it perfectly, but doesn't is not bigger than this and fits like this. It's like, it kind of feels like 3D printing a little bit. It's
Hmm.
like I want this really specific piece to be machined exactly like so with like blank. Um, we kind of have we kind of can do that now. It's such a cool time to be a biochemist. Like, cause
Yeah.
that was such a wall. Like I remember
Such a
like
fundamental like limitation. Yeah.
yeah, it was such a wall, and then they just solved it all in like a couple years. It's wild. Um. Anyway, thank you so much for joining us. We
Yeah, thank
are
you.
out of time now.
Okay. Mhm.
Join us next week at 9:30. Thank you. Oh, we there's a whole bunch of stuff in the chat that we didn't read. I guess the next step is to ask the model how it arrived at the structure to find all the little rules we missed. That would be neat. Think you would need to be more specific on what kind of IR. Yeah, we will bring this topic back again. Bye.
Yep.
Thank you.
I,
Thank you for coming. We really appreciate the chats.
yeah. Have a good night.
Good night. See you next